Protein localization, process whereby proteins accumulate at their correct site of functionality. And proteins can localize anywhere in the cell, depending on what their function is. If it localizes to the cytoplasm, it might be part of the cytoskeleton. Transcription factors need to work in the nucleus, and so their final destination resides in the nucleus. Proteins need to function in organelles sometimes, and so, for example, if you're localizing to the mitochondria, you might be a part of the electron transport chain. And just like we've seen before, receptors need to localize to the cell surface. There's many ways in which proteins reach their correct destination, and the cell puts a lot of energy in. The high degree of organization we talked about, cytoskeletal elements, are designed to help proteins reach their destination of interest. And one pathway we're going to talk about, as an example, is the secretory pathway. This is the pathway by which proteins reach a pathway by which proteins reach cell surface, and it happens through the endoplasmic reticulum to the Golgi to cell surface. So returning back to an extremely simplified diagram, here we see a cell with a nucleus here, and this is the cell surface. And so zooming in on this area, we can see that this is again the nucleus, this is outside of the cell, and here we are seeing the endoplasmic reticulum, Golgi, and then vesicles. And so the secretory pathway to the cell surface is described as this. Proteins are transcribed, translated, properly folded, and they end up in the lumen of the endoplasmic reticulum. They then get placed in these vesicles, transported to the Golgi, and the Golgi then sends them to their location. And here we can see vesicles are heading to the cell surface. If the mitochondria is considered the powerhouse of the cell, then you could call the Golgi a sorting or packaging plant, because depending on where a protein is supposed to end up, the Golgi will send it to different locations in the cell. Now, this process might seem very clear here, but there's a lot of energy and a high degree of signaling that's required for proteins to get to their correct location. And so let's look at just this area right here, how proteins that end up in the ER signal themselves to be entered into the Golgi complex and started on the secretory pathway. So like I said, we're zooming in here. As you can see, this is the ER lumen, and this is the beginning of one of these vesicles here. And so what we see is that proteins that end up in the lumen not only have sequence for their function and are folded properly, but they have an exit signal. So encoded in their amino acid sequence is a signal that will help them localize to the right location. So once they're in the endoplasmic reticulum lumen, there are these cargo receptors on the membrane of the endoplasmic reticulum, which, here, let's grab a pen. These two will bind together, and cargo receptors and proteins will then form these vesicles that then get sent through the Golgi complex. And so the main concept we want you to take away here is that protein amino acid sequence is not just for formation and for proper folding and for correct functionality, but can also be to make sure that the protein localizes to where it's supposed to be. And another cool thing is that mutations now not just affect the function of the protein, but also where it can end up. So, for example, if we were to have a mutation in this protein's exit signal, what would happen is that it could no longer bind with the exit signal on this cargo receptor because the sequence here is different. And you would see that these receptors no longer have protein associated. And so this green protein is no longer able to participate in this secretory pathway. However, other proteins are not affected. Or what about mutations in the actual cargo receptor itself? If the area that recognizes proteins is mutated, it can no longer recognize any proteins, and so the vesicles that get sent through the secretory pathway are empty. So definitely keep in mind that there's an exit signal in some proteins that help decide where it's going to go. and that there are proteins within the endoplasmic reticulum that will recognize and understand how mutations in the exit signal will affect localization versus mutations in the exit signal of the cargo receptor. If there's something you're interested in, there's a very cool course, Bio 315 Human Cell Biology. This discusses all the practical aspects of how cells organize and how proteins and molecules cooperate together in some common mechanisms that the scientific community is aware of. For example, proteins, there's many complicated aspects that are involved in proper functionality, folding, localization, just as we talked about. But there's also very cool aspects in how the cell dedicates a lot of energy towards progressing through the cell cycle, which is very cool. A lot of cool topics like this, with very cool applications such as how drugs work and how cancer signaling is different than healthy cells. So if you're interested in what we've talked about here, definitely check out this course. I highly recommend it. And that brings us to the end of the section on what cells look like. Just as a reminder, we talked about nuclear organization and cellular organization.